So, as part of your syllabus, today we are going to learn the basic concept of linkage and crossing over. And in the coming classes, we'll coming to know about the applications of linkage and crossing over. That means how the chromosome maps, linkage maps, that means genetic maps are constructed. What are the problems related to it? Myself and other lectures will be dealing with the application part. So today we are going to only learn the basic part. So the today's topic is linkage. What is linkage? Linkage is the phenomenon of certain genes staying together during inheritance through several generations without any change or separation due to their being present on same chromosome. Same chromosome. In simple terms, we can define linkage as this: the tendency of genes to remain together during the process of inheritance in the same or original combination as present on the chromosome. So, what exactly this is? So, I have shown here one chromosome, and there are different loci, about 11 different loci are present. Each locus contains one gene. So, these are all genes. Okay? What happens during the process of inheritance? That means if the same chromosome is transmitted to the gamete, from there into the zygote, and then into the organism, what happens? All the genes present on the same chromosome in the same combination. That means the first locus there is gene 1, second locus there is gene 2, third locus there is gene 3. So they are transmitted from one generation to the second or another generation in the same combination. Okay? And this process is called as linkage, and all the genes present and all the genes present on the same chromosome are called as linked genes. Okay? So thus we can define it as simply as this. The tendency of genes. This is the tendency of genes. What? To remain together. To remain together. That means these are not separated. To remain together in their original combination. That means 1 is followed by 2, 2 is followed by 3, like that. In their original combination from one generation to another generation during the process of inheritance. Why this happens like this? Because they are present on the same chromosome. And such a phenomenon is called as linkage. Okay. So I have a string. To which I have added certain beats. So what happens when I take the string with me? All the beats that are present on the string also come along with the string. Likewise, when the chromosome is having certain genes, along with the chromosome, all the genes in the same order, in the same combination, they are transmitted to one individual, from there into his germinal cells, from there into the zygote after fertilization, from there into the next generation. And all these are called as linkage genes, and this phenomenon is called as linkage. And this phenomenon of linkage, due to this linkage, Mendel's independent assortment is not possible. So when we are dealing with the critical review of Mendel's laws, we have said that where there is linkage, there is no possibility of what we call independent assortment. Thus, this is another exception for the Mendelian principles of inheritance, where independent assortment is not possible. So, if you can't remember, I have posted the video already, critical review of Mendel's laws. You can go through that once again. And coming to the discovery of linkage. So, two scientists, namely Bateson and Punnett, while experimenting on the sweet pea, Lathyrus rhodomonas, they have discovered this phenomenon, called as linkage. But they did not know what exactly this phenomenon is. They did not mention the term linkage even. Okay, they don't know anything about linkage, but they are the pioneers in discovering this phenomenon. After that, while working on Rossiter melanogaster, Morgan in 1910, he is behind the concept of linkage along with Castle. So Morgan and Castle are the two scientists who proposed the theory of linkage. And let me explain you now the process of this thing, discovery of linkage. So Bateson and Punnett were working on the sweet pea. Okay, the sweet pea here they have gone for a dihybrid cross. In this dihybrid cross, they have taken two characters. One is flower color. Flower color is said to be purple. Some books it is written as blue. So you can write it down as purple or blue or both. And the recessive is said to be red, purple and red. And the second one is here. Flower color is dominant over purple or blue, flower color is dominant over red color. And the second character that I have taken is the shape of the pollen. Shape of the pollen. These pollen are of two types. One is long pollen, the other is round pollen. Like this. The long pollen is dominant and round pollen is recessive. Okay. So thus, here, Bateson and Punnett in 1906 described a cross in sweet pea where failure of gene paints to exhibit independent assortment takes place. And the characters under study are flower color and shape of the pollen. The flower color is either purple or blue, which is dominant over red color, and the shape of the pollen is either long, which is dominant, or red, which is recessive. Okay. Now he has crossed. Okay. Now they have crossed two plants differing in these two characters, two pairs of characters, hence we call it as a diamond cross. One is having blue flowers and long pollen. The other is having red flowers and round pollen. Red flowers and round pollen. We know blue is said to be dominant. Capital B, like this. Long is said to be dominant. Capital L, capital L. And red and round are said to both are recessive. Double B, small b, small b, small l, small l. So thus, what they have taken is, they have taken in the parental generation a blue long one and crossed it with a red round one. And obviously, as blue is dominant over red, we can get blue or purple color. As long as dominant over round, we get long one in the F1 generation. And now they have gone for the F1 cross. So this F1 cross is not general cross. It is a test cross. It is a test. So what do you mean by test cross? Where the unknown genotype, this unknown genotype is obviously dominant one. So here heterozygous is said to be dominant. Is crossed with a double recessive parent. A double recessive parent. So here the double recessive is sorry, here it is small, small. Okay. It is crossed with the blue long one, which is heterozygous, F1. It is crossed with red, which is double recessive homozygous parent. So this is not the genotype, small b, small l, you write here. So when they have observed that, can anybody tell me what is the test cross ratio for monohybrid cross? Test cross ratio for monohybrid cross.
1:1, sir.
Very good, excellent.
1:1, sir.
Very good. For dihybrid cross?
1:1:1:1, sir.
Very good. In case of monohybrid cross, there are two phenotypes only. So it is 1:1. In case of dihybrid cross, there are four phenotypes. 9:3:3:1 means four phenotypes. Thus we get only four ones here. Okay. And this is a dihybrid test cross. The F1 cross is a dihybrid test cross. So they should get 1:1:1:1. But surprisingly, they have got 7:1:1:7. They have got 7:1:1:7. And this phenomenon is called by them as gametic coupling. Okay, I'll explain you about this later. And they have gone for the second experiment. They've gone for the second experiment. In this experiment, they have taken, sorry, this is not the case. Just a second. Okay. Here they have taken the blue one and the round one, the red one and the long one. So blue flowers and round pollen, red flowers and long pollen. These two were crossed. Blue means capital B, round pollen, small l, red, small b, and what happens? Long pollen, capital L. So this gives rise to one kind of gamete, capital B, small l only. This gives rise to capital B, capital L only. When these two are crossed, we get capital B, capital L, small l, which are said to be again blue and long. Okay, so thus in the F1 generation, they got the blue long ones. What happened is they have again for F1 cross, which is again a dihybrid test cross. The genotype here is small b, small l. And they have crossed the blue long one of F1 individual, of F1 generation, with a double recessive parental genotype, red round one. And if it is a test cross, what they should get? In dihybrid cross, 1:1:1:1 should be the ratio. But the ratio is 1:7:7:1. And they call this as gametic repulsion. The first one is called as gametic coupling, and this one is called as gametic repulsion. In the gametic coupling, what you observe is the parental combinations are more and recombinants, new combinations are less. In case of repulsion, the parental combinations are less and the recombinants are more in number. So they have, after finding these results, they have explained this phenomenon like this. So let us take the first genotype, capital B, capital B, capital L, capital L. And the first genotype of the second genotype of the parental generation, small b, small b, small l, small l. So can anybody tell me what is the genotype of the gametes here? Genotype of the gamete from the capital B, capital B, capital L, capital L one.
Capital B and capital L, sir.
Very good. For this one, small b,
Small b
Small
and
B and
small l.
small L.
excellent. So how many types are produced? Only one type from this one and only one type from this one. When these two are crossed, the combination is capital B, small b, capital L, small l. Okay. Now it has to produce gametes. F1 individual heterozygote produces gametes. So what kind of gametes it produces? Can anybody tell me? What are the four
Capital
different
B, small.
First one, capital
Capital
B,
B, small b,
capital B,
capital L,
small
capital L.
No. How is it
Sir,
possible?
Capital B,
capital B, capital L,
capital L, capital B,
small
small
L,
l,
Very good.
and
small L,
small
capital
b,
B.
capital L, and small b, small l.
Very good.
Both
Excellent.
recessive.
And from this double recessive parent, only one type, small b, small l. So somebody said capital B and small b. So you please remember this fact. Whenever you have doubt, you draw and take one chromosome and draw them like this. So capital B is on one side, on one chromosome, small b on the other chromosome. During the formation of gametes, we know that chromosomal number is reduced to half. That means one chromosome goes to this side, the other chromosome goes to this side. That means capital B here, small b here. So capital B gets separated, small b gets separated. Likewise, if we take capital L and small l, both will not come to the same gamete unless otherwise there is non-distinction we have discussed while reviewing the critically reviewing the Mendel's laws. So capital B, small b, because they are for same character, because they are present on two different chromosomes, they never come to the same gamete. Capital and small l, they are present on the same locus of two different chromosomes, but they can never come because both the chromosomes are separated. So this thing you have to remember always. So what happened here is, here in the parent, in the F1 hybrid, this is the F1 hybrid, there are four different types of gametes, capital B, capital B, small b, capital L, small b, small l. And this, each one of this, I represent it like this now. So capital B, capital L, capital B, small b, capital L, small b, small l. These are the four gametes of the F1 hybrid. And there is a double recessive parent which gives rise to small b, small l gamete. Simple. Now this process, this one, capital B, capital L, small l. This one, capital B, small l, small l. This one, small b, capital L, small l. And this one, small b, small l, small l. Thus we get four different phenotypes where parental combinations as well as recombinants in test cross are same, equal in number. Why they are same in number or equal in number? Because the number of gametes produced having capital B, capital L, let us say they are 100 in number. So that capital B, small l also will be 100, 100, 100. That means they are equal in number. That is the reason why if we get 100 plants each, the ratio is always 1 is to 1 is to 1 is to 1. So the number of gametes that are produced here for each genotype is same. Okay? But what the scientists say here is from this genotype capital B, capital L, small l, after F1 cross has taken place, the parental generations are more in number and new combinations, recombinants are less in number. Why is it so? Again, we go for these two combinations, capital B, capital L, capital B, small b, capital L, small b, small. And for these two, that means parental combinations, blue long, red round, to come in more number, the gametes with blue long should be more in number. The gametes with red round should be more in number, and they should be less in number. So that when they fertilize this one, the recessive gamete with small b, small l, we can get 1:1:7. So what these two scientists are saying here by this experiment that during the formation of gametes from the heterozygote of F1 generation, the gametes with parental combinations are said to be, parental combination of genes are said to be more produced. They are produced in more number, whereas the gametes with new combination, blue with round, red with long, are produced in less number. Why this is so? So this phenomenon is called by them as coupling. Why this has happened here? Because they say that in the in the parental generation, when the gametes are formed, the combination is capital B and capital B, and here the combination is small b, small l. And what is happening here? Capital B and capital L are more in number because this is the combination that is seen in the parental generation. Here, small b, small l are more in number because that is the combination seen in the recessive parent. And they say that both the dominants, capital B and capital L. In other words, for blue color and long pollen are staying together. They are staying together during the process of inheritance. They are staying together during the process of inheritance. Likewise, small b, small l, that means gene for round flower, gene for red flower and round pollen are staying together during the process of inheritance. Very less recombinants are being produced here. And this staying together of dominant genes, capital B, and recessive genes, small b, is called by them as coupling. It is called as coupling. So why this actually happened? At the time they don't know, but we can now say that there are two chromosomes. Here there is capital B, here there is small b. Here there is capital L, here there is small l. What happened now? Because they are present on the same chromosome, the same combination is passed to the next generation, which we call it as linkage. Because these are present on the same chromosome, small b, small l, the same combination is passed on to the next generation. So this staying together of the genes present on the same chromosome is called as coupling by Bateson and Punnett. And let's see the second reaction, the second experiment, where the recombinants are more in number and parental combinations are less in number. So what happened here? Let's see again. Here, what is the genotype of the gametes of this parent? Can anybody tell me?
Capital B, small l.
Excellent. Capital B, small l. What about this one?
Small b, capital L.
Small b, capital L. So that is the genotype of the gametes. So when these cross, capital B, small b, capital L, small l will be the genotype. Now coming back to the heterozygote, which is same as in the previous experiment. So this produces how many? Four different types of gametes. Capital B, capital L, capital B, small b, capital L, small b, small l. And it fertilizes the gamete containing small b, small l. So when all these are equal in number, the ratio will be 1 is to 1 is to 1 is to 1. But the ratio is 1 is to 7 is to 7 is to 1 for this experiment done by Bateson and Punnett. That means, what do you mean by this? The gametes with capital B, small b, capital L are produced more in number than the gametes with capital B, capital L, both dominant genes together, small b, small l, which are produced very less in number. Why is this so? Bateson and Punnett say that as in this cross, here the parental combinations of this are said to be passed on to the next generation. Here also the parental combination is capital B, small b, capital L. So because this is the parental combination seen in the gametes, okay, this is the combination that is seen in the gametes of the parental generation. Even in the heterozygote, during the formation of gametes, the same combination of the parents is said to be followed. Capital B, small l is produced more in number, small b, capital L is produced more in number. So this phenomenon, how we can call this, they have called this as repulsion. Why? They say that capital B, capital L should be one combination, small b, small l should be another combination. But both the dominant genes are repelling each other. Both recessive genes are repelling each other. So that capital B, instead of combining with capital L, it is combining with small l. It is combining with small l. Likewise, small b, instead of combining with small l, because it is repelling it is combining with capital L. So this phenomenon where the dominant genes and recessive genes repel each other is called by Bateson and Punnett as gametic repulsion. Okay, how this happens? We can say that here there is capital B, small b, capital L. This also can happen. So, if because this type of combination is more in number, they are saying that capital B repelled capital L and it combined with small l. Small b repelled capital L, small l, and combined with capital L. So thus, because this combination is existing there, what happened? Gametic repulsion has taken place. But they did not know anything about this, and hence they published this as coupling and repulsion hypothesis. Okay? They just called it as hypothesis. They did not call it as a theory. Why they did not call it as a theory? Because they could prove what has happened. They could explain what has happened, but they could not explain why it has happened like that. They could not explain why it has happened like that. So it is just imaginary theory is called as hypothesis. Okay, and this is the what we call first discovery with respect to linkage theory. Next, this is all we have discussed. The second one for linkage is that of Sutton. So Sutton is a scientist. He has given his view. He also did not explain in the form of a theory. And he said that Mendel said that there are factors. Mendel said that there are factors factors are responsible for characters. Okay? And afterwards, we have discovered genes, and we say that factors are nothing but genes, and genes are responsible for characters. After that, we could discover that how the genes are inherited from parents to offspring, no one knows. But the phenomenon of fertilization, when it is said to be observed under microscope, they have seen that only the nucleus from sperm is transferred to the ovum during the process of fertilization. The nucleus is carrying chromosomes and the ovum is also carrying chromosomes. That means for them, chromosomes carry characters. Okay? So according to Mendel, genes are responsible for characters, but physically, genes are not getting transported. When they are observed under microscope, chromosomes are getting transmitted from parents to offsprings. And this we call them as physical basis of heredity. Chromosomes are considered as the physical basis of heredity because we can see what is happening. What is happening during the process of heredity? Something is transmitted from the parents to us. So physically we can see what? Not genes, but chromosomes. So they say that chromosomes are responsible for the transmission of characters. So which of these is true said to be true? That is what Sutton's question is. And by his imagination, and not just imagination, by his understanding of this thing, he's found that genes are responsible for characters, okay, and characters are more in number. Characters are more in number. But we say that chromosomes are carrying characters, but the chromosomes are less in number. So there are only 23 pairs of chromosomes. That means 46. But how many characters one person have? He can have nearly 1,000 or 2,000 or 5,000 or 10,000 characters. If you say one chromosome carries one character, we can have only 46 chromosomes so that we can carry only 46. Even they are pairs, so that there are 23 pairs and we can carry, these chromosomes can carry only 23 chromosomes. Then what about the remaining characters? Who is going to determine them? Who is going to transmit them? So they said that as the number is not correlating, here this would be like this. There are chromosomes for characters, there are genes for characters, but chromosomal number is less, characters number is more. So what this scientist certainly saying, chromosome carries more number of genes. Chromosome carries more number of genes. Then the problem is solved. But he could not prove it. He could not prove it. Then there, he along with Bovary proposed chromosome theory of heredity in 1902. Okay, in 1902, Theodore Bovary observed that proper embryonic development of sea urchins does not occur unless chromosomes are present. So, for the heredity to take place, chromosomes are required during the process of fertilization. They are the ones that are transmitted to the embryo. That means zygote from there to the embryo. So for embryo to get developed, according to him, chromosomes are required. The same year, Sutton also observed the separation of chromosomes into daughter cells during meiosis. So what we are saying, during the process of gamete formation, both the parental and maternal and paternal chromosomes separate. And this separation of chromosomes into daughter cells during meiosis was observed by Walter Sutton. Putting together these two things, they developed the theory called as chromosomal theory of inheritance. We can also call it as chromosomal theory of heredity, which identified chromosomes as the genetic material responsible for the inheritance of the Mendel's factors. This is what Sutton and Bovary proposed. Chromosomal theory of heredity or chromosomal theory of inheritance. According to them, chromosomes are the physical basis for heredity. Next, By combining all these and also by uh, doing some experiment on Drosophila, while doing experiment on Drosophila melanogaster, Thomas Hunt Morgan, along with Castle, came up with this phenomenon called as linkage. And this linkage, this theory is called as theory of linkage. Why we are calling it as theory? Because it was proved by Morgan. Okay, how we proved? We'll see later. Next, he says that genes are linearly arranged on a chromosome like the beads on a string. So according to him, if we say take one chromosome, here there are genes which are linearly arranged on a chromosome like the string. When we take a string and add beads onto it, okay, what happens? The beads are linearly arranged on the string. Likewise, if we compare the chromosome with a string, the beads can be compared with the genes can be compared with the beads according to Morgan. Next, they also said that genes present on the same chromosome, genes present on the same chromosome, this combination are inherited together. They're inherited together. So this simple phenomenon is given the name linkage. That means because they are linked to each other, this is called as linkage, as simple as this. Next, the distance between the linked genes, the distance between the linked genes in the chromosome determines the strength of the linkage. The strength of the linkage depends on the distance between the linked genes. This I will be explaining in the coming slides. So this is what the theory of linkage as proposed by Morgan and also Castle. So you have to note down these 3 points. First point is they are linearly arranged on chromosomes. Second point is they are inherited together. Third point is the distance between linked genes is determined by the strength of linkage. That means there is a relationship between the strength of the linkage and distance between the linked genes. These 3 you have to remember whenever something question which of the following with respect to linkage theory as proposed by Morgan or Morgan and Castle is said to be wrong. That means these 3 will be given and another will be given as wrong. So when you understand the entire concept, then only you can answer such big questions. Okay. Next, another scientist, he proposed a cis-trans arrangement of the genes. This is called as Haldane's concept. Haldane told us about the cis arrangement of genes and trans arrangement of genes. As I have said before, while Bateson was experimenting on sweet pea, they have observed 2 phenomena, coupling phase and repulsion phase. In coupling phase, both the dominant genes are on the same chromosome, capital B, capital L, small, both the recessive genes are present in the same chromosome. In repulsion phase, what happened? Capital B repelled capital L and combined with small l, capital B, small l. Small b repelled capital L, small l, and it combined with capital L. So this is the combination. So by placing this like this, we see that dominant genes are present on one chromosome, recessive genes are present on one chromosome. This arrangement is called as cis phase. And here, the one and one recessive are present on each chromosome, on the same chromosome. And such a phase is called as trans phase. And now, this is called as, in order to remember the experiments of Bateson and Punnett, cis phase is also called as coupling phase, and trans phase is called as, can anybody tell me what this phase can be called as? Trans phase?
Repulsion phase.
Repulsion phase. So remember, cis phase is equal to coupling phase, where both dominant genes are present on one chromosome. This is one pair of chromosomes, both dominant are present on one chromosome, both recessive are the other chromosome. Likewise, on the same chromosome in another individual, the combination is like this: one dominant and one recessive. Here also one dominant and one recessive. This is what proposed by and then later. And coming to the types of linkage. So we can divide the we can have three types of classifications. First classification is based on the crossing over. Second classification is based on the chromosomes, type of chromosome involved in linkage. And the third one is on the basis of genes involved. Okay. Let's see. On the basis of crossing over, there are two types: complete linkage, incomplete linkage. On the basis of chromosomes involved, there are two types: autosomal linkage and allosomal linkage or sex linkage. And the third one is coupling phase, repulsion phase, which we already have seen. Let me explain the other two. So complete linkage. What do you mean by complete linkage? The genes located in the same chromosome are inherited together, not just from father to son, but to the coming all generations. Why? Because there is no crossing. So due to the absence of crossing over. When there is crossing over is absent, then what happens? There is complete linkage. But there is no single individual, deployed individual on the face of the earth where there is no crossing over taking place during the formation of gametes. So thus, it is a rare phenomenon. But some scientists reported this phenomenon in male Drosophila. So all the genes
Sir,
that
what
are
is
present
crossing
here,
over?
we are going to discuss it after this one. Okay? You just write it down like that. So when, okay, I'll explain this here also. Here the same, the dominant genes are here. All dominant genes are one chromosome, all recessive genes are another chromosome. So what happens? During the formation of gametes, they are separated. That means this comes to one gamete and this comes to another gamete. Then what happens? All the dominant genes are present here. In movies, we see father is Prabhas and son is also Prabhas. In the latest Jawan movie, Shah Rukh Khan is said to be father and Shah Rukh Khan is said to be son. Is it possible? Same to same. It is possible same to same. What happens? When all the genes carried by all the 23 pairs of chromosomes by Shah Rukh Khan are said to be dominant, and all the genes carried on the 23 pairs of Gauri Khan are said to be recessive. Then the child will be purely, totally Shah Rukh Khan like. But that is not possible, the scientists say. Why? There is a phenomenon called as crossing over, where the chromosome gets cut here. It gets a cut here. And after the breakage, this part of the chromosome, this part of the chromosome is exchanged with uh, this part of the chromosome. What happens? This comes here and this comes here. Now, what is the combination now? Previously, small a, small b, small c, small d, small e, smaller, capital A, capital B, capital C, capital D, capital E, capital F. And because there is a phenomenon of exchange of chromosomal segments between a pair of homologous chromosomes, which is called as crossing over, now the new combination is like this: capital A, capital B here, a, small b here. So previously, all small a, small b, all recessive genes are present. Now two dominant genes were added. That means Shah Rukh Khan's dominant genes for these two loci. Let us say one is for his nose, the other is for his eyes. Previously, this combination is present with other one. Now this gone to this one. So the combination is said to be different now. Okay. So this exchange of gametes. So this is the one that he got from his father. This is the one that he got from his mother. Now what is happening? When he is producing gametes, he is crossing it, crossing over taking place in the gametes, and a new combination of genes is being sent to his or her son or daughter. That is what we are all doing. Every organism is doing. So because of this phenomenon, what happened previously, A, B, C, D, E, F, all dominant genes will be transmitted to the next generation. Then that is called as complete linkage, because all the genes present on the same chromosome are inherited together in the same original combination as being present on the same chromosome. But this is not happening here, because there is a phenomenon called as crossing over. So when there is a phenomenon called as crossing over, the genes are not inherited together. When the crossing over is absent, they are inherited together, and this is called as complete linkage. So when there is complete linkage, see, this is capital A, capital B here, small b here. Even in the next generation, capital A, capital B, small b is the same combination, because there is no crossing over. They just are getting transmitted as it is. And such a phenomenon is called as complete linkage, which is not possible. It is a rare phenomenon. And when crossing over is involved, what happened? New combinations, as we have seen, dominant gene with recessive combination, recessive gene with dominant combination. New combinations are produced. We call these as recombinants. We call them as recombinants. So when the recombinants are formed, as was observed by Mendel during the process of independent assortment, there is 9:3 to 3:1 ratio. 9 and 1 are said to be parental combination, round and yellow, wrinkled and green. And 3 and 3 are new combinations or recombinations, round and green, wrinkled and yellow. Why they are said to be round and green and wrinkled and yellow? Because they observed that they have followed a different pattern where the genes responsible for those characters are present on different chromosomes. And even when they are present on the same chromosome, independent assortment is possible because of crossing over. So when there is crossing over, there is no same pattern following to the next generation. And such a linkage is now called as incomplete linkage. So genes present on the same chromosome have a tendency to separate due to crossing over, which produce recombinant progeny beside the parental types. And such a linkage is called as incomplete linkage. Okay. This is the same thing that was said to be observed by Bateson and Punnett. Second one. Okay. Second one is to be based on the type of chromosome involved. Linkage is of type two types: autosomal linkage and allosomal linkage. That means we have 22 pairs of autosomes and one pair of sex chromosomes, XX in female, XY in male. So all the genes present on the autosome are inherited together along with the autosome only. And such a linkage is called as autosomal linkage. All the genes present on allosomes are said to be linked. Because they are present on X chromosome, they are X-linked. Because they are present on Y chromosome, they are Y-linked. And such an inheritance, such a linkage is called as allosomal linkage, based on the genes where they are present. If they are present on autosomes, it is autosomal linkage. If the genes are present, the linked genes are present on the X and Y chromosomes, it is called as autosomal linkage. And third one, we have already explained you about the coupling and repulsion phases, cis and trans arrangement. Now, there will be in some questions, in some examinations, there is a question again on linkage groups. So, what a linkage group? Let's see. A linkage group is a linearly arranged group of linked genes which are normally inherited together. So, I have how many pairs of chromosomes? 23 pairs of chromosomes. So, all human beings are having 23 pairs of chromosomes. That means if one chromosome, the first chromosome is having a number of genes. So this will form one linkage group. All these are called as linked genes. But the other pair is also having genes. So this one also should be called as another linkage group. Thus, in diploid individuals, as the chromosomes are in pairs, we can say that there are 46 pairs of 46 linkage groups. But actually, that is not the case. Why? Because these are the first pair of chromosomes. On the first locus, there is a gene for eye color from paternal side. And on the maternal side also, on the first locus, there is a gene for eye color. So, whether it be black or brown, both genes are responsible for only one character. So, with respect to character, though there are alternate forms, but the character is only one. So, we consider only one chromosome out of a pair when we count the linkage groups, but not the other one. So, we can make it in simple way. There are 46 chromosomes. The haploid number is 23 and the linkage groups is 23. As simple as this. So linkage group is equaling in a diploid individual. Linkage group equals to the haploid number of chromosomes. Okay. So if there is a question on linkage groups, you can simply if there is some question like this is asked, there are 72 chromosomes or there are 36 pairs of chromosomes in dog. What is the number of linkage groups? Then what will you say?
36, sir.
It is 36 because we have to take the haploid number only. Thus, in human beings, the linkage groups are 23. In Drosophila, there are 8 chromosomes, 4 pairs. Thus, we have 4 linkage groups. But there is a small difference with respect to Drosophila and human beings also. In Drosophila and human beings, the male are said to be having 22 autosomes which are paired and same, but the last one is said to be X and Y. So, out of these 22 pairs, there are 22 linkage groups plus 1X because the linked genes present on X are not same as the genes present on Y chromosome. These are X-linked and these are Y-linked. And there is 1. Thus, male is having in Drosophila and also human beings 24 linkage groups. Whereas in female, there are 22 pairs of autosomes which are same. Thus, we have 22 linkage groups here with respect to autosomes, and allosomes are 2X chromosomes. Both are same, so there will be only one linkage group. Thus, there are 23 linkage groups. So there is a small difference with respect to the number of linkage groups when there is a difference in the morphology of the sex chromosomes. Okay. And coming to the strength of linkage. So what actually what we call influences the strength of linkage? So there is a chromosome on which there are certain genes A, B, C, D, E, F. Okay. There is a classroom. There is a teacher here. There are so many benches. Okay. And a number of students are sitting. Okay. And this is the first bench, this is second bench, this is the third bench, this is the fourth bench. Whom do you feel out of these 4 benches that are close to the teacher? The first bench or the last bench? Obviously,
Sir.
the first bench only. Even the lecturer's concentration will be on the first bench only. Okay. Likewise, here the strength of linkage between the teacher and students is more when the student is close to the teacher. If you keep on coming to my staff room, if some student with so many doubts, he or she keeps on coming to my staff room, asking me so many doubts. What happens? There is a bond developed between the student and the teacher. Okay. If you don't come at all to meet me, what happens? I don't know even your name. That means the bond is said to be very less. Likewise, what is making what is influencing the bond between the teacher and the student? The distance. The more the closer you are with your father, with your mother, with your teacher, with your friends, the more the strength of this bonding called as linkage. The more the distance you are, the more the distant you are, what happens? The strength of linkage decreases. So here, capital A and capital B are very close to each other because they're side by side. That means when crossing over takes place, it may take place here. It cannot cut between two adjacent genes because for it to get separated, the breakage should occur somewhere that is far from the telomere. Okay, and the segment is, if it is said to be very small segment, it cannot take part in exchange and fusion. At least this should be somewhat larger segment so that it can be broken, it can be exchanged. So when this is said to be long enough, a long segment is required to be broken. All the genes present here in this segment are said to be close to each other. So that we say that there is more strength between these linked genes. They cannot be separated even by crossing. But when the distance increases, when the distance increases, what happens? When the wife and husband are there, when they are close to each other, they do not hear any words that are said by others. Their bond is said to be close. When they hear some words about wife, if the husband hears, or if the wife hears something about the husband, what happens then? They stop talking to each other. There occurs a distance between them. Then what happens? Someone may come and break their bond. That will lead to divorce. Likewise here, also, what is happening? The distance is increasing between A and E, or between A and C, between A and D, between A and F. The distance is gradually increases. As the distance is gradually increasing, the crossing over frequency may also increase. In that case, these two, A and C, may be broken and they are separated. And they are no more inherited together because C is separated from AB. They are no more inherited together. That means the linkage is said to be, the strength of the linkage is said to be decreased. So here, what we can say is, the strength of linkage. The strength of linkage depends on what? It depends on the distance between the linked genes. The distance between the linked genes. Okay? What actually happens is, when the strength increases, when the distance increases, the strength decreases. When the distance decreases, the strength increases. Thus, it is indirectly proportional. The strength of linkage is indirectly proportional to the distance between the linked genes. That means when the distance increases, the strength decreases due to crossing over. When distance decreases, as there is no crossing over, the strength between the linked genes is to be more. This you have to remember. It is not directly proportional, it is inversely proportional. Hope you have understood. And there are certain factors affecting linkage. What the scientist says, with increased age, the linkage decreases. The strength of linkage decreases. And even the temperature decreases the strength of linkage. And also the X-rays. So these are the different factors that affect linkage: age, temperature, and X-rays. Then what is the significance of linkage? When there is linkage, there are no new combinations, only parental combinations are produced. So it reduces the chances of formation of new combinations. That means same original combinations of the parents are simply being transmitted to the next generation. If we can say that there is a Ongol cow which is said to be strongly built with more number of milk genes, and if the same without any crossing over, the same combination is transmitted to one generation to another generation, what happens? We can get all the newborn babies with the same strong genes combinations, strong gene combinations. Thus, new combinations are produced by linkage. Next, it helps the keeping the parental, racial, and specific traits together. So the parental combinations or the combinations that are seen in the bases are said to be kept together due to complete linkage, due to linkage. It is also useful in maintaining the good character of newly developed variety. So the newly developed variety, if it is said to be not undergoing crossing over, that is passed on to the next generation due to complete linkage. And linkage plays an important role thus in determining the process of hybridization. This is what the significance of linkage is. So hope you have understood linkage. Any doubts?
No, sir.
Okay. So in tomorrow's class, we'll be dealing with the second, because it's already 10 o'clock, 9:50, we are nearing. Tomorrow we are going to discuss crossing over or recombination. So any doubts, you ask me. Otherwise, we'll close the class now. Thank you.